Across the United States, plasma centers open each morning beneath bright fluorescent lights. Donors arrive for different reasons. For some, the compensation helps cover groceries, fuel, or an overdue bill. This is source plasma collected for manufacturing, not whole blood intended for direct transfusion. Machines separate the pale yellow plasma and return red cells and other cellular components to the donor's body. What leaves the center as one compensated donation may then be frozen, tested, pooled, and separated into medicines used to treat immune deficiencies, bleeding disorders, and other serious conditions. The donor receives a relatively modest payment, while the plasma enters a global industry built on scientific expertise, industrial processing, scarcity, and profit. That system saves lives. But it also raises an uncomfortable question. How did a renewable part of the human body become one of modern medicine's most valuable raw materials? To understand how blood entered medicine, return to 17th century Europe, when physicians barely understood either its circulation or its dangers. After William Harvey described circulation in 1628, experimenters asked whether life itself could be transferred between bodies. In 1665, Richard Lower successfully transfused blood between dogs. 2 years later, Jean-Baptiste Denis transfused animal blood into human patients, believing its supposedly gentle temperament might calm mental illness. Some recipients survived small doses, others became gravely ill, and one patient died after repeated procedures. The controversy brought restrictions that pushed transfusion out of European medicine for generations. It returned in 1818 when British obstetrician James Blundell performed the first successful human-to-human transfusion to treat postpartum hemorrhage. He established the crucial principle that human patients required human donors. Yet doctors still could not explain why some human transfusions saved lives, while others caused deadly reactions. Blood had become medically useful long before it became medically predictable. The mystery began to break in Vienna as the 20th century opened. In 1900, Austrian physician Karl Landsteiner noticed that when blood serum from one person was mixed with red cells from another, the cells sometimes clumped together. He realized that human blood was not uniform. In 1901, he described 3 groups, A, B, and C, later renamed O, based on the antigens carried by red cells and the antibodies present in plasma. A 4th group, AB, was identified afterward by Alfred von de Castello and Adriano Sturli. This discovery explained why some transfusions succeeded while others triggered deadly reactions. Doctors could now test donor and recipient blood before combining them. For the first time, blood became something medicine could classify, match, and manage rather than gamble with. Landsteiner's work, later honored with the 1930 Nobel Prize, did more than make transfusion safer. It turned blood into a standardized biological resource. Scientific compatibility solved one problem, but another remained. Fresh blood clotted quickly, forcing donors to stay close to recipients. In 1914, researchers demonstrated that sodium citrate could prevent coagulation. And in 1916, Francis Rouse and J.R. Turner developed a citrate glucose solution that preserved blood for several days. World War I turned those laboratory advances into military necessity. In 1917, U.S. Army physician Oswald Hope Robertson established blood depots near the Western Front. Group O blood was collected in glass bottles, refrigerated, and transported toward casualty stations in insulated ammunition boxes. Wounded soldiers no longer always had to wait for a suitable donor to be found beside them. Blood could be collected before a battle and delivered where casualties were expected. This was more than a new medical technique. It was the beginning of a supply chain. War had transformed transfusion from an intimate exchange between 2 people into an organized system connecting anonymous donors with strangers. By the time the next World War began, blood had moved from battlefield improvisation into civilian infrastructure. In 1937, physician Bernard Fantis opened a blood preservation laboratory at Chicago's Cook County Hospital and popularized the term blood bank. During World War II, mass donor campaigns taught civilians to present blood as a patriotic gift to anonymous strangers. In 1941, the American Red Cross began collecting blood for the U.S. military. While dried plasma, far easier to store and transport than refrigerated whole blood, became a familiar symbol of wartime medicine. After the war, hospitals and nonprofit organizations expanded these networks, turning voluntary donation into a civic duty. The donor was not selling blood, yet the system surrounding each donation was never costless. Staff had to recruit donors, test samples, process components, refrigerate units, discard unsafe material, and deliver products before they expired. Blood entered medicine as a gift, but keeping that gift usable required an increasingly complex economy. The next transformation occurred when medicine learned that a unit of blood did not have to remain whole. Centrifuges could separate blood into red cells, platelets, and plasma, allowing each patient to receive the component needed. Red cells restored oxygen carrying capacity. Platelets helped control bleeding. Plasma supplied proteins involved in clotting and circulation. Then researchers went further. During World War II, Harvard biochemist Edwin Cohn and his team developed cold ethanol fractionation, a method that separated plasma proteins according to physical properties. Albumin became valuable for treating shock and maintaining blood volume, while other fractions later yielded immunoglobulins and clotting factors. One donation could support several therapies instead of one transfusion. This made blood more useful, reduced waste, and expanded treatment. But it also changed its economic identity. Once plasma could be divided into standardized medicines, the value of blood no longer ended with the bag. It multiplied inside laboratories. That multiplying value created a new kind of donor economy. Unlike whole blood collection, plasmapheresis removes plasma while returning red cells and other cellular components to the donor. Because the body replaces plasma faster than it replaces red cells, donations could occur more frequently, giving commercial centers a repeatable supply of raw material. In the United States, companies increasingly paid people for source plasma intended not for direct transfusion, but for manufacturing medicines. The payment was officially compensation for time and inconvenience, yet its economic pull was obvious. Cash attracted people who needed immediate income, while repeat visits helped factories maintain enormous plasma pools. This did not make every donor exploited or every company predatory. It did, however, create a market unlike the civic blood bank. One system asked citizens to give blood as a gift. The other paid them to return regularly, turning the body's capacity to regenerate plasma into the foundation of a scalable business. The same efficiency that multiplied plasma's value also multiplied its danger. Clotting factor concentrates transformed life for people with hemophilia. Instead of receiving plasma transfusions in hospitals, they could inject a small vial at home. But each batch could be made from plasma, pooled from thousands of donors. Before reliable screening and effective viral inactivation, one infected donation could contaminate medicine distributed to many patients. During the 1970s and 1980s, blood products and transfusions transmitted hepatitis viruses and HIV on a devastating scale. In the United Kingdom, more than 30,000 people were infected through contaminated NHS blood or blood products, and the official inquiry attributed more than 3,000 deaths to the disaster. The catastrophe was not caused by pooling alone. Dependence on imported plasma, delayed responses to known risks, inadequate warnings, and institutional secrecy all deepened the harm. A system designed to make treatment abundant had made contamination scalable as well. The disaster forced medicine to rebuild blood safety layer by layer. Donor interviews and eligibility rules became stricter. while every donation was increasingly screened for HIV, hepatitis B, and hepatitis C. Laboratories introduced sensitive antigen, antibody, and later nucleic acid tests, shortening the dangerous window between infection and detection. Manufacturers of plasma-derived medicines added heat treatment, solvent detergent processing, filtration, and other steps designed to inactivate or remove viruses. Plasma could also be held in quarantine until a donor returned and tested negative again. Meanwhile, recombinant clotting factors reduced many patients' dependence on proteins extracted from pooled human plasma. No single safeguard could eliminate every threat, but overlapping defenses made transmission dramatically rarer in well-regulated systems. The industry had learned its lesson. Efficiency could never substitute for traceability, transparency, and precaution. Trust did not return because blood products became risk-free. It returned because safety stopped depending on one test or one institution. Today, that business supports a global dependency. In a May 2026 presentation, the FDA cited 2023 figures showing that U.S. centers collected roughly 50 million liters of source plasma, about 70% of the global supply used for plasma therapeutics. Much of it comes from compensated donors, who may return repeatedly because plasma replenishes relatively quickly. Their donations feed factories that fractionate huge pools into immunoglobulins, albumin, and other medicines used by patients far beyond the United States. This system has expanded access, but it has also concentrated risk. If American collections fall during a pandemic, economic disruption, or regulatory change, shortages can appear in countries that do not collect enough plasma themselves. The contradiction is difficult to ignore. Treatments for immune deficiencies and other serious conditions depend partly on people willing to exchange a renewable part of their bodies for cash. The donor sees one payment. The global market sees a strategic medical resource. That dependency raises an uncomfortable question. How voluntary is a donation when the payment is needed for groceries, rent, or fuel? Paid plasma collection is a regulated exchange, and donors participate willingly. Plasma regenerates, eligibility is screened, and the procedure returns blood cells to the body. Yet consent does not erase economic pressure. A person with savings can decline an appointment. Someone facing bills may feel less free to walk away. Companies do not create that poverty, but their business model can benefit from it by turning financial need into a dependable stream of repeat donors. At the same time, eliminating compensation could shrink supply and threaten patients who rely on plasma-derived medicines. The moral problem, therefore, has no simple answer. Payment can provide both immediate relief and essential medicine. But when global health depends on existing economic hardship, the supply chain is revealing an inequality it did not invent. Once plasma leaves the collection center, however, its price does not rise through profit alone. Each donation must be identified, tested, frozen, and traced. Approved units are shipped to specialized fractionation plants, where large pools are separated through repeated changes in temperature, alcohol concentration, filtration, and purification. Manufacturers must remove or inactivate pathogens, verify potency, fill sterile containers, and satisfy regulators before a medicine reaches a patient. Some plasma may be discarded. Factories, skilled labor, cold storage, clinical research, and distribution all add cost. This complexity explains why a donor's modest payment cannot be directly compared with the final price of an immunoglobulin vial. Yet it does not erase the central imbalance. The person supplying the irreplaceable biological material is compensated once, while companies create continuing value by transforming millions of donations into specialized therapies and long-term contracts. The body provides the resource. Industry controls what happens to it next. For patients, however, these products are not abstract units in a market. A person born with a primary antibody deficiency may be unable to produce enough protective antibodies and may need regular immunoglobulin infusions to prevent repeated, potentially serious infections. Others depend on plasma-derived treatments for bleeding disorders, liver disease, shock, or specific immune and neurological conditions. Their need gives the supply chain its urgency. Production cannot expand instantly when demand rises. Plasma must first be collected, tested, pooled, fractionated, purified, and released, a process that can take months. A shortage, therefore, cannot be solved simply by raising a factory's output next week. Hospitals may postpone treatment, restrict doses, or prioritize patients for whom alternatives are limited. This is why plasma commands such value. Its price reflects industrial complexity, but its power comes from something more fundamental. For the patient waiting at journey's end, the finished product may be irreplaceable. Governments have answered that dependence with competing philosophies. The World Health Organization urges countries to build blood and plasma systems around voluntary, non-remunerated donors and pursue national self-sufficiency. In that model, donation itself is separated from direct payment, although the processing and sale of the resulting medicines may still involve commercial companies. Yet principle collides with scale. Collecting enough plasma for industrial fractionation requires large volumes, regular donors, specialized centers, and sustained public investment. Countries that cannot produce sufficient plasma-derived medicines must import them, often from markets supported by compensated donation. This creates a paradox. A nation may reject payment at home while its hospitals use therapies made from plasma collected for money abroad. Neither system is morally pure. The unpaid model may preserve public trust, but without sustained recruitment and public investment, it can struggle to generate sufficient volume. The paid model can expand supply while concentrating collection among people most attracted by cash. The global market survives by linking these contradictory choices across national borders every day. Behind national policies stands another concentration of power. Only a limited number of companies can fractionate plasma at industrial scale. Building a plant requires enormous capital, specialized equipment, regulatory approval, and years of validation. Many major manufacturers therefore operate vertically integrated networks, linking collection centers, testing laboratories, refrigerated transport, fractionation plants, and international distribution. This structure can improve traceability and coordinate supply, but it also creates bottlenecks. If one large facility fails inspection, suffers contamination, or shuts down for maintenance, patients far beyond its borders may feel the shortage. Competition authorities have recognized the danger. In 2011, the U.S. Federal Trade Commission required divestitures before allowing Grifols to acquire Talecris, arguing that the merger could reduce competition in several plasma-derived therapies. Blood may begin with millions of individual donors, but the power to transform it into medicine is concentrated in far fewer corporate hands, shaping access and prices worldwide today. The fragility of this concentrated system became visible during COVID-19. Lockdowns, fear of infection, staffing problems, and reduced mobility kept many donors away from collection centers. Because plasma-derived medicines can take many months to manufacture, the full effect did not appear immediately. Falling collections entered the pipeline like a delayed shock, threatening future supplies of immunoglobulin and other therapies long after the first missed appointments. Countries dependent on imported plasma suddenly discovered that contracts could not create biological material that had never been collected. The crisis also exposed a deeper limitation. Plasma cannot be stockpiled indefinitely as a complete solution, and new fractionation capacity cannot be built overnight. Some governments responded by encouraging domestic collection and reconsidering self-sufficiency. The pandemic had turned an invisible supply chain into a question of national medical resilience. Rebuilding national supply proved far slower than lifting a ban. England resumed collecting plasma for medicines in 2021, but donors, testing systems, storage contracts, fractionation capacity, and regulatory approvals still had to be assembled into one functioning chain. Only in March 2025 did NHS patients begin receiving medicines made from UK-donated plasma for the first time in a generation. Even then, self-sufficiency remained partial. NHS Blood and Transplant aimed to meet roughly 30 to 35% of national immunoglobulin needs from domestic plasma from 2026, not eliminate imports overnight. The lesson extends beyond Britain. A country can recruit donors yet still lack enough collection sites, specialized processing, or resilience when demand rises. Replacing dependence therefore requires years of investment, public trust, and reliable participation. Governments cannot manufacture that biological foundation by decree alone. Blood begins inside individual bodies, but medical sovereignty depends on an entire industrial system built around them. Biotechnology offers a path beyond this dependence, but not a complete escape. Recombinant DNA technology already allows manufacturers to produce clotting factors without extracting them from donated plasma, reducing infection risk, and giving many people with hemophilia alternatives to plasma-derived concentrates. Newer engineered proteins, monoclonal antibodies, and gene therapies may further reduce demand for particular components. Yet immunoglobulin presents a much harder challenge. It is not one simple protein that a factory can copy repeatedly, but a vast mixture of antibodies created by thousands of human immune systems responding to different infections and vaccines. Researchers can manufacture individual antibodies for specific diseases, but they cannot yet reproduce the full diversity of pooled human immunoglobulin at the scale patients require. Technology may therefore replace some products while making the remaining plasma more valuable. The industry's future may be less dependent on blood, but it is not yet free from the human body. The future of plasma medicine will depend partly on how this market is governed, and partly on whether biotechnology can eventually reduce dependence on human donors. Donors need clear information, careful health monitoring, reasonable donation limits, and compensation that does not disguise risk or exploit desperation. Patients need diversified supplies, transparent pricing, and public stockpiles capable of absorbing temporary disruptions. Governments can invest in domestic collection without pretending every country must build an entire fractionation industry alone. Regional partnerships may spread cost and reduce dependence on a single exporting nation. Companies, meanwhile, should be judged not only by how much plasma they process, but by how reliably they protect donors and maintain access to essential therapies. None of these reforms removes the central contradiction. Human plasma is both part of a person and raw material for medicine. A responsible system must treat it as both, because ignoring either identity produces harm at global scale. Return to the plasma center where this story began. The donor rises from the chair, presses cotton to his arm, and leaves with a payment that may help solve one immediate problem. Behind him, the pale yellow bag enters a system of freezers, laboratories, factories, contracts, and hospitals. Science has transformed blood from a mysterious fluid into a classified, divisible, and globally traded medical resource. That transformation has saved millions of lives. It has also allowed inequality, corporate power, and national dependence to become embedded inside the same supply chain. The question is no longer whether money belongs in medicine. It already does. The question is whether the people whose bodies sustain that medicine will receive protection, dignity, and a fair share of its value. Blood may leave the body as a donation or a paid exchange, but once it enters the market, humanity must decide whether it remains a gift or becomes merely a product.